Hello students, welcome to
VEDANTU JEE English. This is Basavraj sir.
This is Gopika ma'am.
We are your
Biology
master teachers for today.
Yes.
So let us, before we start, let me tell you about myself. My name is Basavraj. I teach botany on VEDANTU NEET English channel.
And I am Miss Gopika. I teach zoology on the VEDANTU NEET English channel. So today we are here to do a small help. That is when you are writing your ICER aptitude test, you have around 15 questions of biology, right?
Right. So we'll be solving some questions, and these questions are going to be your previous year questions and also the expected questions. So if you have any doubt regarding anywhere, just tell us and let us know in the comment section. So before we start, quickly smash that like button, and we'll start with the small, high weightage units now. Right, ma'am? Can we start? Let's not waste any time.
Yeah, sure. Okay, so students, I know most of you, if you're coming from a PCM background and if you're a PC like B, then you don't have anything to worry because you would have already prepared for all the chapters. But imagine you're from a background who does not want to study bio, but you want this 15 marks. Then which are the chapters you need to focus in botany and zoology? We'll tell you right now.
So when it comes to botany, I'll tell you what exactly where the questions will be repeated every year. Students, listen to me very carefully first of all. The first important one is going to be the cell structure and function. Every single year, one or two questions will keep on repeating from here. Especially, do you see this cell cycle and cell division? Especially that meiosis 1 prophase. From there, question has been repeated multiple times. Then we have your plant kingdom, animal kingdom. Not many questions, so you can not disregard, but less attention there. Anatomy, you can expect some questions. Then plant physiology. In your plant physiology, main questions will come from C3 cycle or C4 cycle, or there will be questions from your glycolysis, Krebs cycle, and electron transport system. That is the location where you need to put more attention to. That is when it comes to botany. Ma'am, when it comes to zoology, human physiology.
Yes. So human physiology is very important, but also I noticed that in the previous years, a lot of questions have come from animal tissues, that is structural organization. Frog is there, so it's very important for you to remember the animal tissues. Other than that, human physiology, all the chapters, questions have come. I think if you're preparing for NEET or if you have prepared for your 11th and 12th, you will be able to answer that. And at the same time, genetics, we have to tell you. Reproduction also, I have seen quite... match the following is there, yeah. Few questions are there, not as much as weightage how NEET has for reproduction. That much I do not see it.
But ma'am, the importance of genetics is very peculiar here because every year one or two questions will come from genetics, and that's for sure. Out of the 15 questions, one question will be from genetics, and it's either going to be from your molecular basis or principles of inheritance. Apart from that, we can expect a question from your ecology. It's given the next page. I'll show you. That is the botany, end of the botany. That is going to be your ecology. Ma'am, ecology, especially the organisms and population, questions have repeated from there. So you don't need to study the entire thing because we know less time is there. Try to focus on the high weightage units first and then go for the other chapters. We would recommend that. Right,
yes,
ma'am?
Yes.
Here.
And also here students, evolution also I've seen very few questions. Evolution, animal kingdom, you can expect maybe one or two questions. But again,
rare, right?
Yeah, it's very rare. So do not waste your time right now. At this point, do not waste your time. Go to the high weightage chapters. Human health and diseases, I have seen a lot of questions from antibodies. So make sure that you're studying immunity part very, very thoroughly with antibodies. Okay. So that's the thing. So if you're starting your preparation now, we have told you from our end. Biotechnology also, I have seen a lot of questions based on your gene and also questions from molecular basis on basis of your experiments. Okay, Frederick Griffith experiments, Michelson and Stahl experiment, all those experiments. Okay, yes, NCERT is the goal again. NCERT,
Exactly.
Read NCERT.
Read NCERT. Okay. So students, right now we would love to focus on some important questions or different concepts which you need to keep in your mind right now. Yes. So we have around 40 questions, 45 questions with us, and we'll be solving this question. We'll be teaching you the concept also along with the question. So follow through it. Any doubt, let us know in the comment section. We will reply to it. And ma'am, can we start with zoology?
Yes, sure.
So we'll be starting off zoology and then we'll be coming to botany questions.
Yes, okay. So students, are you all ready? I cannot see the attendance mark. Come on, this is how you welcome biology, yeah? So come on, mark your attendance and let's start the zoology questions. Very easy questions. But yeah, it is previous year questions as well as some questions that we expect. Okay? Yes, ecology part also is important. Okay? Ecology part also is important. You can expect questions, especially from your Yes, so let's start with the first question. I hope question is clearly visible. Yes, question is visible. Okay. So during COVID-19 pandemic, SARS-CoV-2 virus mutated multiple times, giving rise to many, okay, mutated multiple times, giving rise to many variants. What is the genetic material of your sarcovirus? That is, again, COVID time, all of you know that your severe acute respiratory infection had happened. That's the reason why people were not able to breathe, right? People had difficulty in breathing and we had to supply maximum oxygen. Many people died because they were not able to get oxygen cylinders. What was the reason? Which was the genetic material behind this for mutation to happen? The options given are single-stranded RNA, double-stranded RNA, single-stranded DNA, double-stranded DNA. One thing all of you should remember when it comes to virus, the biggest catch about virus is that it has the ability, right, to multiplicate fast or to mutate fast. It can form maximum variants. It can form maximum variants. I can see many of you are telling answer A. So yes, I totally agree. Option number A is correct answer. That is single-stranded positive RNA was the reason why, even though we put Covishield and Covaxin, when the second time a variant came, right, JN1 variant came, people were scared. That fear was because virus can always form its varieties, and that varieties we cannot treat with just one vaccine. Very good, those who told answer A. Okay, very good. Now, next question. These are all previous year questions, so don't worry, your previous year questions are, done properly here. Osmoreceptors are sensitive to changes in ionic concentration and volumes of body fluids. Which among the following best describes the function of these osmoreceptors? Osmoreceptors are, osmoregulation is there. That means ionic balance, salt and water balance. That is osmoregulation. Now, osmoreceptors, osmotic pressure also has to be maintained in the blood. It has to be maintained throughout in our body. Okay, whatever it be, water concentration, whatever, if salt is there, that salt concentration also has to be maintained. Here they are talking osmoreceptors are sensitive to changes in ionic concentration and volumes of body fluids. Which among the following best describes the function of osmoreceptors? Okay, can you all try? Can you all try? Single strand means what? Like this. If you go here, this is single stranded, this is double stranded. Okay, two strands are there which are complementary to each other and they wound around each other. Single strand means only one strand is there. Double strand means two strands are there which are going around with each other. Okay, yes, yes, got it. Okay, now they stimulate hypothalamus to facilitate the release of antidiuretic hormone or your vasopressin. Other name is vasopressin. Okay, and increase the water reabsorption. They stimulate pituitary gland to facilitate the secretion of mineralocorticoids to adjust the changes in the mineral composition of the body fluid. They stimulate atrial wall to release natriuretic factor to reduce vasodilation and reduce blood pressure. They stimulate renin production to induce vasoconstriction and increase blood pressure. What do you think is the answer? What do you think is the answer? Yes, give it a try. Give it a try. What do you think would be the most correct answer? Those who have studied chapter, those who know something about your renin, RAS mechanism, ANF mechanism, ADH mechanism, maybe can give it a guess. So look here, osmoreceptors are sensitive to ionic concentration. That means your salt and water balance. Volumes also, this means H2O if water reduces. So the most appropriate answer is they stimulate hypothalamus to facilitate the release of antidiuretic hormone or vasopressin, right? Vasopressin, and increases the water reabsorption. Whenever water is less, automatically your ionic concentration also will decrease. So automatically everything is getting affected. So the answer is A. Very good, very good. Awesome, awesome. Answer is A. Okay, so everything is interrelated. Water balance reduces, then ionic concentration also will decrease. So everything will keep decreasing. Now, interneurons play an important role in the execution of spinal cord mediated reflex action. Where are these interneurons located? Now, what is interneurons? All of you have studied neurons. We know that we have sensory neuron, right? We have motor neuron, and we have association neuron, or we can also call it interneurons, right? So that means motor neuron is there, sensory neuron is there, association neuron is where both are there. Some sensory, some motor is there, right? Some sensory and some motor is there. So here they are asking where is the position or where is the location. Option is grey matter, white matter, dorsal root ganglion, muscle spindle. Now we know that in your entire spinal cord, when you study your reflex action, you study that there is a head-shaped structure which has a grey matter and a white matter. Okay, a grey matter and a white matter. In that also we know that white matter is always made up of the axons. That means the myelin sheath, right? The myelin sheath given out is myelinated axons will be little whitish in color. That means the myelin sheath is white in color. So we remember like that, white matter because of the axons. Grey matter is because mainly of your cell bodies and your dendrites. So what do you think the answer is going to be? Among A and B, okay, okay. Yes, reflex arc is deleted, but this question is a part of your, when you study the neurons, type of neurons, this is there. Okay, this is there. Okay, what's the answer? All of you are telling me answer A. Correct? See, look here. In your textbook, if you open your neural control and coordination, you will see this. See, the cerebral cortex is referred as the grey matter due to its greyish appearance, and the neuron cell bodies are concentrated here, giving the color. Who is concentrated? Cell body or the cyton of a neuron. Okay, cyton of a neuron. The cerebral cortex contains motor areas, sensory areas, and a large region that are neither clearly sensory nor motor in function, and they are called as association neurons or association areas. But now the catch is responsible for complex function like intersensory association, memory, and communication. So this is what they're talking about. Okay, this is what they are talking about. Clear? So here they're talking about what? Association areas. That means where some sensory is there, some motor is there. It's like a between portion, right? Some of you are not there, not here, right? Some of you are not in class, nor you're studying. So that middle portion, that is called as your association area. So that is what they're talking about. So it's not from the deleted topic, it is there in your chapter. Now, next one question from Animal Kingdom. Organism in which of the following phylum are triploblastic, acoelomate, and have bilateral symmetry? Okay, triploblastic, okay, acoelomate, and bilateral symmetry. Students, this is for IAT exam. Okay, ICER aptitude test. 15 marks comes from your biology. 15 questions will come from your biology. So we are practicing that here. Yash, I hope it's clear. Yes, all of you try reading this. Organism, which of the following phylums are triploblastic, okay, acoelomate, and bilaterally symmetrical? You know that the only phylum that where you will see acoelomate, not pseudocoelomate. The only phylum which is pseudocoelomate is Ascalminthus. Okay, pseudocoelomate is Ascalminthus. You can remember it as VA. Vucheria and Cyclostoma and Ascaris. This is a way to remember the example. The answer is Platyhelminthes. Platyhelminthes. Most of your options are always A, right? Yes. Which one of the following statement is true? Okay, students, don't have the notion that everything will be A. I saw many videos where every student is like, okay, answer everything A. But you should also understand the concept, because you know it's not always going to be A, right? Okay, maybe most of it will be A, but somewhere again might not be A. Okay, yes. Which one of the following statement is true? Let's see. Blood is a connective tissue which does not secrete collagen. I totally agree, because when we study your animal tissues, we study something about fibers, and in fibers we study about collagen and elastin. We study about collagen and elastin, And in animal tissues, it is clearly written that blood is the only one that does not give out fibers, that does not have fibers. So collagen also won't be there, elastin also won't be there. So the first one is correct according to me. Okay, correct. Now, tendons are a type of dense irregular connective tissue. Wrong. It is a type of regular connective tissue. Regular connective tissue, and the way to remember this, is LA-DRI. Okay, L stands for loose connective tissue, A stands for adipose tissue, A stands for areolar tissue, D stands for dense connective tissue, R stands for, wait, I'll write it down. Okay, this is the example for your loose connective tissue. Loose connective. Okay, so this is how you'll remember. This is adipose and this is areolar. Got it? Yes, easy. Students, if you leave the bio session, how will you get 60? How will you get 60 marks? You need full 60 marks, no? Yes. So you have to learn a little bit of bio. I know some of you might be like, no ma'am, I have never studied in my life. So how do I study? But just do the high weightage chapters. Previous year questions you're solving in this session only. So you do not have to worry about that. Okay. Are we good to go? Yes. Yes or no? Tell me fast. Okay. And yes. And here DRI, this is actually to remember dense connective tissue as regular and irregular. Under regular, you have tendons and ligaments. Okay, tendons and ligaments. This is type, students. You have to study this in your animal tissues. Dense connective tissue is two types, regular and irregular. Okay, regular and irregular. Under regular, example is tendons and ligaments. So here the question is, tendons are a type of dense irregular connective tissue. Wrong statement, regular. Adipose tissue is an example of, tell me, all of you look here and tell me, adipose is an example of which type of connective tissue? Try to tell me. Adipose is a type of which type of connective tissue? Look here and tell me. La I have written. L stands for loose connective tissue. Example is adipose and areolar. Look at this and try to tell me. Yes. Triploblastic, that means 3 germ layers are there. Ectoderm, endoderm, mesoderm. Ectoderm, mesoderm, endoderm is there. When all 3 layers are there, they are called as triploblastic. Acoelomate means there is a wall, there is a cavity between the body wall and the body wall. So that cavity is called as coelom. When coelom is not present, it is called as acoelomate. When fake coeloms means the coelom is in the form of pouches. That is called as pseudocoelomate. Very good, Kumarvel. Very good. The answer is loose connective tissue. Students, look here. Adipose, areolar. Adipose tissue is an example of fluid connective tissue. No, it is an example of loose connective tissue. Okay, got it? Yes. Okay, now saliva is secreted by salivary gland. Saliva is secreted by salivary gland and it's an exocrine gland, not an endocrine gland. So what is the answer? What will be the answer? I have scratched out everything. Which is the correct answer? Yes, try telling me which is the correct answer. Faster, faster. Yes. Which is the correct answer? Is it A, B, C, D? Okay, I told you tendons are a type of dense regular connective tissue. Adipose tissue is an example of loose connective tissue. Saliva is secreted by salivary gland. A is the answer. Very good. Very good. Okay, awesome. A is the answer. Now next question. Oh my god, such a big question. Can you all see clearly? Yes, Yash, if you are a JEE aspirant, you do not have to listen to this lecture. This is for IAT. It is written clearly, no? IAT biology. Okay, don't take tension. The diagram represents an enzyme, its substrate and potential inhibitors. Okay, now look here, you will see some inhibitors are there. one active site is there. This is what you need to focus. Okay, active site is only what you need to focus. Got it? Now, which one of the following combination is the best pair of competitive inhibitors for the enzyme? Let's try to understand this. Competitive inhibitors means, imagine there is a lock and key. Okay, lock and key. This is the lock, this is the key. And normally we will open it, right? Instead of the key, I make a fake copy of it, or I make something very similar to a key with a wood piece or something. That becomes an inhibitor. So, the lock is the enzyme, the key is the substrate. Enzyme substrate complex will be formed. But instead of the key or instead of the substrate, if an inhibitor comes, that fellow becomes a competitive inhibitor. Competitive inhibitor will be the same shape of a substrate and they'll go and bind with the active site. Okay, so look here, active site, what is the shape of the active site? Yes, I know all of you know the answer. What is the shape of the active site? Yes. What is the shape of the active site? What is it? V-shaped or this oval shape, box shaped? What is the shape? V-shaped, right? It's V-shaped. So if you look here, your P has a V shape and your S has a V shape, which can correctly go and bind in this active site. Okay, don't think about your allosteric site. You do not have to take tension about it. You only have to focus about the active site. Always remember, active site is what you need to focus. So the answer is going to be P and S, because P also can correctly bind there, S also can correctly go and bind there. Is this clear? Is this clear? Now, match the entries. Very good. Triangular. Yes. Match the entries in column 1 with their function described in column 2. Students, when you are studying biology, even if it's the first time you are studying biology, make sure that you study animal tissues. Lot of questions have come from animal tissues. Okay. And I hope all of you marked your attendance by smashing the like button, because that's the only way for me to know how many students were there in the class. Okay. Awesome. Now, match the entries. Column 1. We have squamous epithelium. Always remember this. Squamous epithelium always help in filtration, diffusion, all that job is done by squamous epithelium. It will be flat, okay, flat cells, okay, yes. Now, look here, squamous epithelium, it serves as a diffusion barrier is there. So, I am going to mark that, okay. Then imagine I don't know anything else, okay, I only know squamous. Imagine I only know squamous. How will I answer? P is 4. If you look here, the only option where P is 4 is option number A. I can blindly answer, but since I am a teacher, I am going to explain you more. Cuboidal epithelium. What is the answer for cuboidal epithelium? The nucleus is at the center of the cell and it helps with secretion and absorption. Cuboidal means, always remember, it will help for secretion and absorption. Okay, secretion and absorption. Yes, it is deleted from your new NCERT. But it was a 2023 question. Okay, 2023 question. I think in your this, yes, you can learn it from PDF. Just go to the animal tissues PDF and you can learn it from there. Okay. Yes, you can also answer ciliated, no problem. Okay, so here Q is 3. I'm giving it 3 because secretion and absorption always cuboidal epithelium. Now, I know ciliated means movement. Movement. Ciliated means, always understand, cilia means hair-like structures. They will always help in moving something, right? Moving something. So, hair-like structure means moving. So, the answer is going to be A. Okay, clear? Next question. When a ribosome encounters a stop codon, okay, in the mRNA during translation, which one of the following will bind to the stop codon? So, if your entire translation is happening, your stop codon is here. After that, one more person will be there to stop this, okay, to stop the translation. Who is that going to be? Who is that going to be? Yes. Who is that going to be? Yes, rho factor, okay, release factor, termination factor, sigma factor. Now, look here, in your NCERT, if you go and read properly, you will know that when the ribosome encounters a stop codon in the mRNA during translation, which one of the following will go and bind with the stop codon? Who will bind with the stop codon? Okay, so here the question is translation. You are all telling me the answer for transcription. Students understand? Remember like this. Transcription, you have to remember rho factor and sigma factor. Okay? Transcription, rho factor, sigma factor. Okay? Now, termination factor. Such a factor is not there. Okay? So, the answer will be what? I removed this, I removed this, I removed this. The answer is going to be release factor. Okay? Release factor. So, the answer is release factor, and look here. Here you can see at the end a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide form of the ribosome. This is in your NCERT. Okay, got it all of you? So it will bind to the stop codon, and this is seen in the translation. Translation. Okay? Yes. Now, next one. For which one of the following disease does the causative agent require the splicing of their hnRNA to generate mature mRNA? Now, let me tell you very clearly, there is two types, right? Prokaryotes, right? Prokaryotes and eukaryotes. Prokaryotes and eukaryotes. Please understand that splicing is a process where I remove the people who are not good for anything. So I have an mRNA, I have a gene. In that gene, there are some people who will help for sequencing, some people who will not help for anything. I want to remove them. That process is called splicing. Removal of introns is called as splicing. Splicing, capping, tailing, all of this is called as RNA processing. RNA processing will not happen in prokaryotes. Okay? RNA processing will not happen in prokaryotes. It will only happen in eukaryotes. So now among this you have to find which is an eukaryote, and the answer is going to be malaria, because all of this will not come. Okay, got it? So eukaryotes, RNA splicing is there. So the answer is malaria. Now, in an island with 10,000 individuals, 4 have sickle cell anemia. Sickle cell anemia is a recessive autosomal disease. Assuming that the locus is Hardy-Weinberg equilibrium, how many individuals in that island are expected to be heterozygous? I know all of you are like, oh, everything is answer A, everything is answer A. I will just blindly answer without understanding the concept. It is going to be difficult. Okay, so try to understand the concept. Now, how many of you know to solve this? How many of you know to solve this? Shall we try solving it? Yes. Yes, can we try solving it? Okay, now let's see. 10,000 is the total number. Okay, 4 people have got sickle cell anemia. So I am going to do 4 divided by 10,000 is equal to 4 into 10 to the power minus 4. Okay, so this is my Q, right? Q square. Q square is equal to 4 into 10 to the power minus 4. So I think I'll do it here. Next page. Okay, look here students. I want to find Q. Okay, first I will find the homozygous, that is the recessive one. So I will find Q square is equal to, I got 4 into 10 to the power minus 4 by doing 4 divided by 10,000. Okay, now Q is equal to square root of 4 into 10 to the power minus 4. How much it will be? 2 into 10 to the power minus 2. Am I correct or wrong? That will be Q is equal to 0.02. Now, can you all find P for me? Can you all find P for me? I found the Q. Can you all find P for me? What is the formula? P plus Q is equal to 1. That's the formula you have to substitute. So now I know the Q value. So I will do P plus 0.02 is equal to 1, or P is equal to 1 minus 0.02. How much that is going to be? How much will the answer be? I want all of you to tell me the answer. Don't sit silently. Is it 0.98? 0.98? Okay, now the question is, I found the homozygous, or I found the recessive and the dominant. Now the question is, I have to find the heterozygous. Very good, Midun, very good. That is, now I have to find heterozygous one. Heterozygous means 2PQ. 2PQ means 2 into 0.98 into 0.02. All of you multiply and tell me the answer. All of you multiply and tell me the answer. After that only we can go forward. What will be the answer of this? What will be the answer of this? Yes? Yes? Yes, try to tell me the answer. 0. Okay, thank you. Who is that? Ruchir, thank you. 0.0392. But do you see that option here? Because we have to remember that it was in 10,000. So you will do into 10,000. Your answer is going to be 392. All of you understood how you got answer A? If you blindly go and mark option A, you will not get the answer. That's not how you should do the question, right? Lot of people are competing, and you know you have less seats, around 1,800 seats in all the colleges. So you have to be sharp, you have to be prepared for whatever it is. So this is the procedure, this is the way you do it. So the answer is going to be A. I hope all of you understood. Okay. If I want to tell you about autosomal and recessive, it's going to take a lot of time because I have to tell you about test cross, back cross and all. So as of now, let's try to focus on what we can do in a limited time. Okay? Yes. Okay, so the answer is 392. Next one. Which one of the following epithelial cells is commonly found in the inner surface of the fallopian tubes? Always remember, inside fallopian tube and your bronchioles, there is something to be, movement should happen. Bronchioles and fallopian tube, always remember this. Even if a question comes differently, instead of fallopian tube, if bronchioles is asked, you should know that both of this, some movement happens. Movement is required. Okay? So movement, who is going to do the movement? Tell me who is going to do the movement. Very good. Very good. Ciliated epithelium will do the movement because they have hair-like structures called as cilia. Right? Hair-like structures called as cilia. They will do the movement. Got it? So the answer is going to be ciliated. Awesome. Some individuals start sneezing when the pollen content is high in the air. Primarily, which Ig isotype will these individuals produce as an immune response? Anytime you see an allergy, okay, allergy, any kind of allergy, pollen allergy or your walnut, peanut, all the nuts allergy, or some people have allergies with silica, dust, asbestos, so many kinds of allergies people have. Whenever you see the word allergy, okay, whenever you see the word allergy, you should always remember about IgE. IgE. Okay, it's less than 1%. IgE. Okay, got it? Clear or not? So, the answer is going to be IgE. Whenever you see the word allergy, always remember IgE, because that is the first person to go and fight the foreign particle. Okay, to show the inflammation that, yeah, you have an allergy. Okay, you have an allergy. Got it? That is the most important thing. Now, I think all of you know about IgA. You will see this in the colostrum, the milk that comes out, right, from the mother to give it to the baby, that will have all the IgA. Okay, now IgM and Ig, if you want to know the number, this is the highest in number. Okay, around 80 to 85% you have IgG. Now, next one. Which of these factors is least likely to cause deviation from Hardy-Weinberg equilibrium? Please understand, Hardy-Weinberg equilibrium, where you study about your p square plus 2pq plus q square, please understand that this formula or this equation is a very ideal situation equation. What do I mean by ideal? What do I mean by ideal? That means all the conditions are favorable. All the conditions are according to the law. That will never happen. If Hardy-Weinberg equation or if Hardy-Weinberg formula has to fail, mutation should happen, genetic drift should happen, gene flow should happen, natural selection should happen. All this, if it happens, Hardy-Weinberg principle will change. So Hardy-Weinberg principle is when evolution does not take place. That means mutation does not take place. So according to this, what's your answer? Yes, very good. Very good, Ruchir. Yes. Please tell me what's the answer. Can you try? So here I am telling you, if mutation happens, it will definitely cause a big deviation to Hardy-Weinberg. Gene flow will make a difference, genetic drift, reduction in population size. A small reduction in population size will not make a big deviation in Hardy-Weinberg principle. So the answer is going to be A. Okay, answer is going to be A. Oh my God, such a big question. Okay, so here basically they're talking about your Griffith's experiment with your Streptococcus pneumoniae. So Frederick Griffith was the first scientist to ever find out that something can get transformed. Okay, so he came up with transforming principle. He did not know that it was DNA. He just knew that something has come from the S strain to the R strain and made the R strain virulent. Virulent means disease causing. Okay, now let's see here. First situation, T1, S strain, heat killed, injected into the mice. All of you please know, S strain is the one that was virulent. Virulent means the ability for causing any disease. Okay, R strain is non-virulent. It will not cause any kind of disease. Okay, it will not cause any kind of disease. Now, I heat killed the S strain. That means I killed it because of heat, extreme heat. Will the mice live or will the mice die? All of you tell me in the chats, will the mice live or will the mice die? I heat killed it. Even though it was virulent, I put so much heat and I damaged whatever I had to damage. So will the mice be alive or not is your answer. All of you tell me the answer, by then I'll go to the next one. S strain, heat kill, R strain, together the mice will die or live. Nucleic acid isolated from the S strain, that is heat kill plus R strain, incubated with RNAs. We all know that the transforming principle is DNA. If I use RNAs, it is going to remove the RNA. Still the mice is going to die here, no? The mice will die here. Right, the mice will? Very good. The mice will live. The first option. Correct. Okay, here the mice will live. Here heat killed R strain, here the mice will die. Because something has got transformed from the S strain to the R strain. Okay. Now here, nucleic acid isolated from S strain plus R strain incubated with DNAs. If I kill the DNA or if I remove the DNA, then definitely the mice will live. Right, because that was the villain. The villain, if I remove, the hero will be alive. Okay. S strain live, injected into the mice, live. Okay, live injected into the mice will die. R strain is non-virulent, so the mice will live. So which is the answer? Which is the answer? The answer is option number A. Any doubts? Any doubts? Tell me, no doubts? Okay, awesome. So here the mice lived, here the mice will die. Here what is happening? Yes. So here what is happening? Here it is RNAs. RNAs no use. RNA is not causing any harm. Okay. So here what will happen? The mice will die. Okay. Here what will happen? The mice will live because DNAs is the villain. Okay. I mean DNA is the villain. We removed it with the help of DNAs. Okay. Here again the mice will die. Here the mice will live. Question is little big, but the answer is very easy. Okay. Now, the diagram given below shows a typical ECG. If an AV node is atrioventricular node, is not functional, which of the following option correctly represents the changes seen in an ECG? Understand, here this is atrial depolarization, we tell. Here ventricular depolarization. Okay, here atrial repolarization. Atrioventricular node does not function. That means the ventricles will have a problem. Ventricular, you know, that here, see, ventricular depolarization will have an issue. Whatever is coming from the atria, it has to go to the ventricle, right? Whatever is there on the top, it has to go down. So that will not happen. So like that, if you look like that, which one do you think is getting affected? Okay, which will get affected? Try to tell me. Very good, Vaishnavi. The answer is option number B. Always you people are telling option number A. Now you see option number B is the answer, because the QRS complex will be absent. This is a ventricular depolarization when AV node is not functional. Okay, that will cause a problem in the ventricle. So the answer is B. The answer is B, not A. Okay, the P wave will sustain longer with smaller amplitude. No, atrial depolarization will happen. Ventricular depolarization will face an issue, because here you can see QRS complex depolarization of the ventricle triggers the main pumping contraction. That is the QRS complex. Okay, there you can see depolarization of atria in response to SA node triggering. Here it is AV node triggering. So whenever AV node is not functional, ventricular depolarization will get affected. Is this clear? So all of you who told A, you're wrong. Answer is B. Okay, B. B also will come as answer. That's why I told you learn the concept. Now, which one of the following physiological function is common between small intestine and the renal tubules? Here your renal tubules, they're talking about your PCT, DCT, okay, Bowman's capsule and your glomerulus, all of those people except your collecting duct. Okay, wait, let's write. We forgot one person. Whom did we forget? Main hero we forgot, loop of Henle. Okay, loop of Henle. So both of them, small intestine and renal tubules, will help in the absorption of glucose. Absorption is happening even in renal tubules, even in small intestine. All of you know, in small intestine, after all the digestion in the stomach, it will go to the small intestine. There absorption will happen. Whatever useless this fellow has thrown, small intestine will take it back. So the answer will be absorption of glucose. Good job, some of you told the answer. Now look here, an E. coli whose DNA is fully labeled by N15 is grown in a medium containing N14Cl. Okay, this is the heavy nitrogen, this is the light. Together, heavy and light together will form the hybrid, because our DNA replicates in a semi-conservative manner. Semi-conservative manner. Okay. Now, try to say this. Try, look at this. What will be the percentage of hybrid N15, N14 and light DNA, that is both N14s, DNA molecule at end of 80 minutes? Assume that doubling time of E. coli is 20 minutes. Always understand E. coli, the replication happens every 20 minutes. Every 20 minutes. So, I know that four replications will happen in 80 minutes. 4 replications into 20 minutes will give me 80 minutes. Okay? Okay? 2 4s are, all of you know, 80 minutes. Now, my first replication, I will always get, okay, my first replication I will get full hybrid. That is 100% hybrid. 100% hybrid. Now, let's say we write it here. Are you all ready to solve this? Hi, Samat. Okay. Look here, in the first 20 minutes, you'll only get hybrid. Okay, that is full 100%. In the second one, what will happen? 40 minutes. Can you try telling what you will get? Students, hybrid will keep reducing because N15 is not there in that content. N14 is there. So always remember, hybrid is going to be less. Hybrid is going to be less. Is this clear? Give it a try. What you will get in 40 minutes? I know all of you are in a hurry to tell the answer, but I want all of you to solve with me. In 40 minutes, I will get 50% hybrid and I'll get 50% light. Then in 60 minutes I'll get 75% light and I will get 25% hybrid. So what will I get in 80 minutes? 80 minutes I will get 87.5% light and 12.5% hybrid. So what's my answer going to be? What's my answer going to be? After 80 minutes, any guesses? Option D. Option D. Again, people are telling option A. They're asking after 80 minutes. Yes, give it a try. You'll definitely get it. I think. Yes. D is the answer. Okay, people are telling D. Okay, and since you are telling D, I will agree to D. Yes, good job, good job. D is the answer. See, look here. After 80 minutes, you're getting 87.5% light and 12.5%. Here the question is, how many hybrid am I getting? 12.5%. How many light am I getting? 87.5%. So the answer is going to be D. Answer is not always A. Okay, answer is D. Did you all understand? Next question. What are the types of immunity acquired by transfer of antibodies to the fetus? Okay, transfer of antibodies to the fetus via the placenta. That means we are giving the antibodies from the mother to the placenta. Two more questions. Okay, we are giving antibodies from the mother to the fetus. Okay, so whenever we are giving a ready-made antibody, mother, that antibody was already there. We are giving that to the baby who is growing, right? Whenever we do that, it is going to be passive immunity because it is already ready-made. You went to a shop, bought a ready-made dress and came. So that means it's already made. You're just going and buying it. That will be passive. But imagine I give you the cloth. You have to design. You have to stitch. It's going to be active. You'll be very active. No, I have to make the design. I have to do this. Think like this. So this is going to be passive immunity. Vaccination of an infant. In vaccination, I'm going to give an inactivated form. In the body will start forming its own antibody. So it's going to be passive and active. Passive and active. Not B, not B, not B. Carefully listen to me. Vaccination in an infant. Vaccination is a weakened antigen. That means I am giving a weakened foreign particle to my body. My body will start forming antibodies against it. Always remember this. So what will be the answer? Okay, what will be the answer? Try to tell me. Yes, very good. D is the answer. So this is passive. When I am giving ready-made, it's passive. When I am giving a weakened antigen and asking my body to make antibody, it's going to be active. Awesome, awesome. So the answer is D. Okay, next question. DASH is a chronic disorder in which alveolar walls are damaged due to the respiratory surface. Alveolar walls are damaged. And let me give you a clue. This happens because of smoking. Extreme smoking causes this disease. Can you guess it? Can you guess which is the answer? Can you guess? D is the answer. Okay. DASH is a chronic disorder in which alveolar walls are damaged due to which respiratory surface is decreased. Okay. It's decreased. Smoking causes emphysema. Answer is B. Okay. Because there is a human protein called as alpha-1 antitrypsin that will get damaged when a person continuously smokes. Okay, so because of this, that person will end up having emphysema. Okay. Yes, Geeta, for percentage, I'll show you. You have to do semi-replication. That means you have to do this. This is the parent. This is the daughter. Like this you have to keep doing. Then only you will understand. Every 20 minutes you have to do this. That means see, in first you will get like this. In first replication you will get like this. That means 2 hybrid. Complete hybrid means 100% hybrid. Okay. Then separate them. Separate them. 1, okay. 2, separate each of them. This is the only way you have to do it. Otherwise after some time you will know the percentage. Then what you do? Add for everything. Yellow color is light. For everything you add light. Now tell me how many hybrids, how many light? Students, yellow color is light. Light. That means N14. Yellow color is N14. Okay, this is N15. Geeta, can you tell me the percentage? Now looking at this, you can identify. I think orange is not very visible. So I'll draw it with green. Students, green color is the heavy one. Okay, green color is the heavy one. I want the answers fast so we can do botany now. Try telling. Okay, look here, all of you. I told yellow is light. So how much light I am getting? I am getting 2 of it. Out of 4, 2 is light, 2 is hybrid. So can I say 50% hybrid, 50% light. Students who are studying this for the first time, I know it's going to be difficult, but students who already know it, yes, very good Geeta, you understood now. So these 2 are hybrid, the ones with green and yellow, these 2 are light. So when you draw the next one, you draw all of this individually. So 1, 2, 3, 4, 5, 6, 7, 8. 8 strands you draw, and in the 8 strands you add light DNA. How much you will get? That time you will get 75, 25. Again you draw 8 to 16 it will become. That time you will get 87.5 and 12.5%. Okay. After some time you will be used to this. Now it will be little difficult. That's why I have drawn this and shown you. It will take a lot of time if I draw the entire thing. But if you want, you can check out our videos on the channel. Every topic is done there. If you need any topic in biology, you can check out our playlist. Okay, my playlist is there on Vedantu NEET English. You can check out. There I have explained all of it. Now, the factors which does not affect the rate of alveolar diffusion. Alveolar diffusion depends on solubility of the gas, how much oxygen and carbon dioxide exchange can happen, thickness of the membrane. Because always understand, gases move from a region of higher concentration to a region of lower concentration. Middle, there will be a layer. Okay, that layer thickness matters. Pressure gradient matters. Imagine 104 and 90 is there. Air will move from a region of higher concentration to a lower concentration. So pressure gradient also matters. Reactivity of the gas will not matter. Very good. Vaishnavi correct. Kumar correct. Answer is option number D. Okay, so we are done with zoology. Yeah, last question. Each hemoglobin molecule can carry a maximum of how many oxygens? Okay. Yes. Yes. Tell me. Each hemoglobin molecule can carry a maximum of how many molecules of oxygen? Very good. Very good. The answer is going to be, if you know the hemoglobin structure, you don't know right now, it will be too much for you to learn. Always know hemoglobin can carry 4 oxygens. Think of it as a bus. Okay, hemoglobin is a bus. In this bus, there only 4 people can go. 4 oxygen molecules can go. Always. Okay?
You can call it as a car only, then.
Yeah, car also you can call, but bus is easier because, I don't know, bus is easier, students.
Car, four-seater, no.
So, 4 molecules of oxygen will always go with the hemoglobin. Okay? Hemoglobin is the bus. Yes. So, that's the end of your zoology questions. I think we did a fast. Okay, so students, if you have any doubt in any topic of zoology, you can go back to our videos and check. Otherwise, you can just practice previous year paper. Okay? Yes? 1 second, I have to
solve. Were they able to solve?
Yeah, they have notion that all the answers are A. So some of them are just doing A for everything. Students, don't do like that. You need proper understanding. Okay? Students, carbon dioxide, you need to carry oxygen, not carbon dioxide, right? Carbon dioxide is to be thrown out. Oxygen needs to be taken to the tissues and cells for cellular respiration. Okay, so oxygen is actually bad for us, but it has an affinity. It has a great affinity. Carboxyhemoglobin, carbaminohemoglobin. But again, is it clear, Padmavati? Okay. Yes. Okay, thank you so much. So that's it. See you all on, I don't know. So if you want biology classes, come there. But yeah, this is going to be, it was a great thing that we came to this channel, JE channel. And thank you so much. I'm meeting a lot of new students. So bye-bye students, take care. If you want small portions, doubt clarification, come to our channel and check, because we do not, time is very short, right, to explain everything and all the chapters. Yes.
So students, my name is Basavraj sir. I'm your botany master teacher. So let's try to solve some questions. So before we start, tell me in the chat quickly, is the presentation visible and you're able to see all the questions clearly? Ma'am, where's the pen, ma'am? No, thank you. Yeah. Yes, students?
4 molecules, just tell, no? Carbon dioxide also 4 students. Hemoglobin structure is like that. Whatever it carries, 4 it can carry. Okay, yes, but it's harmful.
Okay, let's solve some botany questions now. One by one, I'll explain you, and then we'll see how it goes. So, for first question is very simple, and all of these questions, students, all of these questions are previous year ICER questions, and they are actually a mixture of questions. Some questions are very direct, such as this question. Some questions are very direct. Some questions are very what do I say? Need to make you think a little. Okay, so we are gone year wise. This is 2023 question. Then we'll go 22. Then we'll go 2019, 21, and 17. Okay. So let's start with the first question. That is going to be your, which of the following statements regarding the seed structure is incorrect? Now, before all of you start bombarding, sir, option number A. Let me explain. Before you start bombarding, sir, option number A is the right answer. Wait, let me explain to you. Which of the following structures, statements regarding the seed structure is incorrect, right? Now, in monocot seed, the membranous seed coat. So, all of you know there is something called as seed coat. Yes, there is something called as seed coat that is fused with the fruit wall called as aileron layer. Let me show you the diagram and you should be able to answer on your own. See the diagram. 1 second. See here. Where do you see aileron layer? This is going to be the structure of your monocot seed. This is the structure of monocot seed. In this monocot seed, do you see that this layer right here? Where is it? Aileron layer is not given here. This is your aileron layer. Here, can you see this layer? Can you see this layer? This is your aileron layer. Now, this aileron layer is not required for separation. Aileron layer is required for nutrition. Aileron layer is required for nutrition, not for separation. Okay. But in some cases, students, in some cases, some of the monocot seeds, the fruit might end up sticking to the seed, but it will not be in all the cases. Okay. Only in few cases the fruit wall, that is the pericarp, might attach, but in the most of the cases it is not. That is, the monocot seeds, the membranous seed coat has fused with the fruit wall. It's called aileron layer? No. It is not called aileron layer. Aileron layer is a nutritive tissue. What is it? It is a nutritive tissue. Next question. See here. Now this was a controversial question. This was a very controversial question from last year. some of them even asking for bonus marks. And if you look at any form of internet, if you go check any videos, everyone is saying option number A. Everyone is saying option number A, but do you think it's option number A? Students, let me explain you why it is not option number A and it is given in wrong in many cases. See, the mitotic drug inhibits the microtubule formation. So there is a drug which is going against formation of microtubules. And if you read NCERT, right now, under prophase, if you look NCERT right now, under prophase, which is the under prophase, if you look at the prophase right now, there it shows formation of microtubules. Remember, formation of microtubules happens in the prophase, not in the metaphase. Although microtubules are attaching to the kinetochore, in your metaphase. But what is the question? The question is saying that drug inhibits the microtubule formation. So microtubes are not forming at all. So if the drug has been applied, the first phase of your mitosis, all of you know the trick, right? That is PMAT. That is prophase, metaphase, anaphase and telophase. The first phase, which is called as prophase, this itself does not happen. So answer is going to be option number C. Not A. It's going to be option number C. Okay. Next question. Which of the following anatomical features of wood can be used to estimate the age of a tree growing in the temperate climates? See, all of you know how to count the number of, how to tell the age of a tree? Does anyone know how to tell the age of a tree? If you look at the leaf, you'll get to know. No. How many of you seen something called as? How many of you know what is something called as annual rings? How many of you know something called as annual rings? Do you know when you cut a tree open, you will see some rings like this. Imagine if this is a tree and you're cutting open the tree, and inside the middle trunk you will see some ring like structures. Have you seen it? Have you seen number of rings? By looking at the bark, no. By looking at the bark, you cannot tell the age of a tree. See, obviously, if your tree is very small, you will tell, sir, this is a small plant. If the plant is grown very tall, you will be like, sir, it is a very tall plant. But imagine you have a tree which all the trees are very old. All of them are very old, probably around 30 years old. How can you specifically tell this is 50 years old, this is 60 years old? By counting the number of rings. They are called as annual rings. Like how you celebrate your birthday, right? Like how you celebrate your birthday every year. Whenever the tree gets older by 1 year, there will be one ring formation here. Imagine there are around 20 rings here. If there are 20 rings here, the tree is that many number of years old. Okay, counting the number of annual rings. Now, the question is little twisted here. Now, the question is asking you, answer we know, it is annual rings. But options, do you see annual rings anywhere? Do you see annual rings anywhere in the answer? No. You do not see annual rings anywhere. Because you need to know how are the annual rings formed. Like how are they even forming it? That is the actual answer. Now listen to me very carefully, all of you. I'll tell you how the annual rings are formed here. So how many seasons do we have? We have summer season, winter season, rainy season, right? Summer, winter, rainy, tell more, no? Come on. What are all seasons do you have? We have summer, winter, rainy, then all of you know spring, autumn, right? So in this some different seasons, when you have spring season and autumn season, there is something called as cambium. So whenever there is high temperature, whenever there is springtime, whenever it is springtime, the activity of cambium increases. The activity of cambium increases in the summertime. in the springtime. And when the activity is increasing, the wood which we get out of it is called as early wood. It's called as what? Early wood or spring wood, also called as spring wood. But there is one more season called as autumn. In the autumn season, what happens? In autumn season, the activity of cambium completely decreases. If activity of the cambium is decreasing completely, the wood which we obtain from it is called as late wood. It is called as what? Late wood. So in case of your springtime, we get early wood. In the case of your autumn time, we get late wood. So whenever there is a autumn, when we have light color, we have one early wood, then late wood, early wood, late wood. So seasonal wise, there will be more and more accumulation of this wood. And obviously one wood, which is your spring wood, is going to be light in color. Autumn wood is going to be dark in color. That is why we have multiple layers of wood there, which gives the characteristic appearance of annual ring. That's how the annual rings come into picture, because of spring wood and late wood. So answer number A. I know this concept is, you need to understand this concept. There is no point in mugging up all this. Okay, where did I keep the pen? Now this is a direct question. This is a direct question from NCERT. Which of the following is an example of genetic diversity? Genetic diversity. Quickly, following example of genetic diversity. Is it option number A, B, C, D? Okay, fine. I'll ask you one more question. How many of you like mangoes? Tell me. How many of you love mangoes? Mango season is coming, right? This is mango season. How many of you love mangoes? Tell me. Vaishnavi, Dhanushri. It's B. Are you sure it's not B? How many of you love mangoes? So I love mango. Okay. So in the market, are there only one type of mango, or we have multiple types of mango? We have only one type of mango, or we have multiple types of mangoes? Different types of mangoes. No, one we have small mango, Alfonso mango. There are different varieties of mango. There are multiple varieties of mango. That variety of mangoes is called as genetic diversity. It is called as genetic diversity. That is 1,000 different strains of mango and 5,000 different genetically diversity of rice. So in rice also we have multiple types. No, we have 30 rupees one, 35 rupees one, 40 rupees one, 50 rupees one. Do we only have one type of rice? No. That different types of options, and we have multiple type of strains, that is called as genetic diversity. Within a genus, within a species itself, we have many varieties. Within a species itself, we have many varieties. And here, can you see this line here, NCERT? Revulfia, Vomitaria is an example of genetic diversity. So in NCERT, students, in NCERT, directly example based questions will come. Directly they'll take one example and they'll ask you. Imagine instead of asking Raufacia, they might ask you mangoes this year. They can ask you mangoes, right? They can ask you mangoes. Clear? Now, or they can ask you, some of you saying option number B. Option number B is species diversity. That is diversity at species level. For example, Western Ghats and Eastern Ghats. Western Ghats, this is the India map, right? This is the India map. This is the India, South India, right? This is Kerala, Tamil Nadu. This side is Tamil Nadu, this side is Karnataka. This is your Western Ghats, this is your Eastern Ghats. In Western Ghats side, we have more number of amphibians compared to Eastern Ghats. That is your species diversity. Now what is the answer? Tell me. Which of the following is an example of genetic diversity? Variation of potency and concentration of respine, which is a toxin produced by the Raufacia. High diversity of amphibians in Western Ghats and Eastern Ghats, that is species level. Greater variety of ecosystems found in your India than in your Scandinavia. A greater diversity of plant species found in your India compared to Central Asia. What is the answer now? Obviously option number A. Option number A is the answer, which explains the genetic diversity. Genetic diversity. Now look at this question. Students, one question, see, in your NCR, when I was looking at your past 3-year papers or 4-year papers, one question has always come from the cell chapter. It could, and specifically cell cycle and cell division. All of you please study this chapter, that is cell cycle and cell division. It will approximately take you 3 hours. It will approximately take you 3 hours to master this chapter. Just 3 hours. Just 3 hours you should be able to understand this chapter, and one question will definitely come from cell cycle and cell division. Okay. Now let's look at the question now we have on our hand. This is a pedigree analysis question. This is a pedigree analysis question. The following pedigree chart shows the inheritance of genetic disorder. Some disease is sent from parent to the offspring. Some disease is being transferred. I2 and I3 are only affected individuals. What is I2? Here, affected individual. Third generation, second one. This is affected individuals. Affected. Which of the following is the correct pattern of inheritance of disorder and genotype of I3? They are asking you the genotype. Genotype of I3. Now, the only way to remember this is you need to know that every single type of disorder, that could be autosomal dominant, autosomal recessive, X-linked, Y-linked, every single disease has certain characteristics. Every single disease has a certain characteristics. And the characteristics for your autosomal recessive, recessive is that it will skip generation. It will skip generation. See here, in this generation disease is there. In generation second, do we have any disease? No. In generation third, we have a disease. So can I tell, since it is skipping generations, since this disease is skipping generation, it is autosomal recessive? Yes, it is autosomal recessive. Now, imagine if this, and since this particular human does not have the disease, but the offspring has it, can they be carrier? Yes, they can be carrier. In that case, they can be heterozygous in nature, and since it's skipping generation, it's going to be autosomal recessive. It is going to be autosomal recessive. Next question. Match the following. Very easy question, all of you. Very, very easy question now. Cell with cytoplasm and no nucleus. They're asking you a cell. They're asking you a cell which does not have any kind of cytoplasm as well as no nucleus at all. Now, what is that thing? That's going to be your mature sieve tubes. Mature sieve tubes has cytoplasm but does not have any nucleus. What about then cell lacking both cytoplasm and nucleus? They do not have cytoplasm, they do not have nucleus also. That is going to be your tracheids. That's going to be your tracheids, because tracheids' main function is conduction of water. Main function is conduction of water. They do not require any kind of cytoplasm or nucleus. Cells containing more than 2 nuclei, that is in the case of tapetum. Tapetum is a type of nutritive tissue. Then we have haploid cell with produced by mitosis, that is going to be your synergids. That's going to be your synergids. Let's see, one is T, yes. One is T, and 2 is going to be R. 2 is going to be R, tracheids, yes. 3 is going to be S, tapetum, and haploid cells is going to be synergids, yes. Next question. Shown below are some of the reactions that occur in the metabolic pathway leading to the complete oxidation of glucose during the aerobic respiration. During the aerobic respiration, the glucose is undergoing multiple steps. The first step is happening in the cytoplasm. That is called as glycolysis. The second step is going to be link reaction. Then we have Krebs cycle. Then we have electron transport system. This is the complete flow chart. What is the flow chart, all of you? First is going to be glycolysis. Then we have your link reaction. Then we have Krebs cycle. And then we have electron transport chain or electron transport system. This is the entire flow. Let me write it big. I think it's not visible. First we have glycolysis. Then we have link reaction. Then we have your Krebs cycle. And finally we have the electron transport chain. This is the entire flow. This is the entire flow. Now you need to identify. Which one belongs to which category. First of all, pyruvate to acetyl-CoA. Pyruvate to acetyl-CoA is getting converted. That does not belong to glycolysis. DHAP, that is dihydroxyacetone phosphate, is getting converted to glyceraldehyde thiazide phosphate. This belongs to glycolysis. Then we have oxaloacetate to citrate, not glycolysis. Fumarate to malate, not glycolysis. Now the second one, pyruvate to acetyl-CoA, that is going to be part of your link reaction. That is part of your link reaction. In link reaction, pyruvate is getting converted to your acetyl-CoA. Then we have oxaloacetate to citrate, that is also part of your Krebs cycle, which happens in the initial stage. And finally we have fumarate to malate, which is also the part of your Krebs cycle. So what is going to be the order? The order is going to be which is the first one, 2 and 1, 2, 1. So option number A, right? Option number A. Next question. A single point mutation in gene X. There is some type of mutation here. In gene X results in the breathing difficulty, one symptom, hypertension, second symptom, as well as partial sterility. So mutation in one gene is causing many different diseases or many different symptoms. Which of the following phenomena best explains the observed phenotype? Very easy. This always I always tell to my students, it is like your mother. In your house, your mother will cook for you. She'll help you in any type of requirements. She'll also clean the house. So mom will do every single work in the house, right? Mom is like always active. That is like your pleiotropy. What happens in pleiotropy, students? In pleiotropy, a single gene is responsible for multiple phenotypic expressions. That is going to be pleiotropy. So what is the answer? One gene is able to extend. One gene is showing multiple phenotypes. One gene, many phenotypes. Pleiotropy. Next question. Consider the following biomes: tropical rainforest, tundra, desert, coastal zones. Choose the most probable descending order of net primary productivity. Very easy question again. First of all, let me tell you what is the meaning of net primary productivity. Net primary productivity is the amount of food prepared by the plants which is available to the animals. I'll repeat again. Net primary productivity is nothing but amount of food produced by the plants which is available to the animals. Now tell me, in which particular regions we will have the highest number of plants? Will it be in tropical, tundra, desert or coastal regions? Where will you have a higher number of plants? Obviously in the tropical rainforest, right? In the tropical rainforest you will have the highest number of plants. That is going to become first. So P is 1, so P should be first. So these two options are gone. Second, do you see more plants in your tundra, desert or coastal regions? See, all of you know, in the case of your coastal regions, we might see some plants, no? In the case of coastal regions, we might see some plants compared to tundra and desert. What is tundra? Tundra region is nothing but very cold region, very cold climate. Desert also, all of you know, very hot. But comparatively coastal regions, we might see some plants, lot of coconut trees and anything. Second is going to be coastal regions. That's going to be PS. So answer is A. Answer is going to be A. Next question. Let X be the perpendicular distance from the centromere of each chromosome to the equatorial plane of human cell. What is the question? They're basically trying to ask you, what is the distance if this is a cell? Let X be the perpendicular distance from the centromere of each chromosome to the equatorial plate. This is the equatorial plate in the middle. This is one chromosome here. This is one chromosome here. Let me. Imagine there is a chromosome here. Chromosomes are here. They're asking you, which of the following stages of the cell cycle is most likely to have the highest average value of X? The meaning is that which chromosome is at the furthest distance from the central plate? Which chromosomes are in which particular phase of cell cycle the chromosomes, that is centromere, is away from the equatorial plane? Clear? See here, in the case of your metaphase, what happens in the case of metaphase? In the case of metaphase, all the chromosomes will align to the equatorial plate in metaphase. Do you think they are very close or very far? Imagine all the chromosomes are on the equatorial plate in the metaphase, right? Everyone is here. Everyone is going to be here. Are they close to the equatorial plate or very far? Obviously, they are sitting on equatorial plate. So the X value will be very low. So B can't be the answer. What about your early anaphase? In see, early metaphase also is gone, because metaphase again they're coming very close to equatorial plate. What about your early anaphase? In early anaphase, all the chromosomes are just separating out. In early anaphase, all the chromosomes are just separating out. See here. All the chromosomes are just separating out here in early anaphase. All the chromosomes are just separating out. They are not very far from the equatorial plane. This is the distance. But in the case of your late anaphase, all the chromosomes are already here. All the chromosomes are very far. In the case of late anaphase. If you look at this particular diagram, can you see? All the chromosomes have come here. In the case of your anaphase, or if you take late anaphase, in the case of late anaphase, all the chromosomes are at the each end. This is the equatorial plate. In the middle, we have the equatorial plate. Obviously, students, the answer is going to be what? The answer is going to be late anaphase, in which all the chromosomes are towards the end. No, nothing will come from out of the syllabus, don't worry. Okay, late anaphase. Now, which of the following is not a sexual reproductive structure? This is very easy question. They're asking you which is not a asexual reproductive structure. So we know conidia of penicillium is which is going to be asexual. Zoospores of chlamydia is asexual. Gemmules in sponges are asexual. But isogametes in the case of your cladophora are sexual gametes. Following is not an asexual. It means they're asking you which one is sexual here. So isogametes of cladophora is going to be sexual. Now what is the meaning of isogametes? In iso means same. Iso means same. Next question. Which of the following is or are produced by the plants during the photosynthesis with far red light? So students, all of you should know there are two types of photosystem. We have photosystem 1 and photosystem 2. Photosystem 2 works at 680 nanometer. Photosystem works at 700 nanometer. Now whenever there is far red light, the wavelength of light is generally more than 680. So whenever there is far red light, right, photosystem 2 will not work. Photosystem 2 will not work at all. So only and only photosystem 1 will work. Yes. And we know photosystem, only when photosystem 1 is working, that is the case with cyclic photophosphorylation. Cyclic photophosphorylation. In cyclic photophosphorylation, only and only ATP is produced. In cyclic photophosphorylation, only ATP is produced. There is no formation of H+ ions. There is no formation of NADPH. Because we know there is no splitting of water also. In case of a cyclic photophosphorylation, the electron is coming back. Okay, only ATP is produced. Next question. Which of the following floral formula represents the zygomorphic flower with diadalphous androecium and superior ovary? First look at the zygomorphic condition. Zygomorphic means percentage sign. So option number B is gone. This is gone because this is actinomorphic. This is going to be actinomorphic with diadalphous androecium. The meaning of diadalphous is that all the androecium, all the stamens are attached into two bundles. All the stamens are attached into two different bundles. Can you see two different bundles anywhere here? Infinite bundles gone. This is also gone. There are only two androecium here and they are not fused. So gone. So can you see here? 9 plus 1 condition. 9 is one bundle. This is separate one more. So all the androecium are separated into two separate bundles as well as your superior ovary. So option number A is correct. Option number A is correct here. Now, the following events are associated with meiosis. They have given you entire list of meiosis. You need to tell me which one is at which stage. Now, this is going to be the most crucial one. This is your entire NCERT. Entire NCERT for meiosis 1. Meiosis 1. Please read this one. Meiosis 1, prophase 1. What happens in your prophase 1? Now, a lot of questions can come from here. But let's focus on the question which we have on our hand right now. They're asking you the exact one by one what is happening. See, in the case of leptotin stage, which is the first stage in your prophase 1, there is compaction of chromosomes continue through the leptotin. What happens in zygotin stage? In the case of zygotin, there is formation of synaptonemal complex. Clear? Now, what happens in your pacotin? In the case of your pacotin, there is this stage is characterized by the appearance of recombination nodules. Okay, that is second one. What happens in the third one? And the crossing over also happens here. Crossing over happens here. Then we have, in the case of your diplotin stage, there is formation of chiasmata. Now you tell me what is happening first. First one was what? Formation of synaptonemal complex. After that there was what? Formation of recombination nodules. Third one was formation of chiasmata, and this is the last one, that is formation of mitotic spindles, which will happen in the case of your metaphase directly. So we just discussed that only 3. Why this is given? No, this is wrong. B can't be first. It has to be 4. This is the answer, I guess. 4 synaptonemal complex formation. Then we have formation of chiasmata. No, 4 1. Here this is the answer. That is formation of synaptic animal complex happens. Then you have appearance of recombination nodules, formation of chiasmata, and formation of spindle fibers. This is the answer, students. Option A is the answer, not B. Not B. A is the answer. Sometimes PPT can make mistake, but we should not. Now, in the standard nomenclature used to depicting floral formula, which of the type of flower is represented by K? So we saw here, right, floral formula in the back somewhere here. So this one is usually your symmetry. This one is going to be your sexual condition. This is bisexual. K is going to be calyx. K is going to be calyx. C is going to be corolla. A is going to be androecium, and G is going to be gynoecium. What is the question? The question says that what is K and this 5, you see, it is inside the bracket. Whenever something is inside the bracket, it means that it is completely enclosed and it is very closed. It is closed. So we know gamo means closed. Gamo means closed. And K is sepals, so gamosepalous. If it was C, and if it was written like this would have been gamopetalous. See, C is corolla. Corolla is made up of petals. Calyx is made up of sepals. Which of the following helps in bacterial motility? Very important question. How can bacteria move? We know bacteria moves, but how? Now there might be confusion between these two one. See, all of you know myotubules is not the answer. Centrioles help in cell division. Centrioles help in cell division. The confusion is between always fimbriae and flagella. Fimbriae as well as flagella. All of us know when it comes to fimbriae, its main role is attachment. Main role is attachment. While the flagella helps in motility. Flagella helps in motility and fimbriae helps in attachment to the substratum. So what is the answer? Answer is going to be D. Now, during light reaction in chloroplast of tobacco plant, where is the proton gradient generated? They're asking you, where is the proton gradient? So before we go to the concept, let me tell you the meaning of proton gradient is that there is a difference in proton levels. And if you know the structure of chloroplast, this is the structure of chloroplast. We have some coin-like structures called as thylakoids. Thylakoids. Entire stack is called as grana, and this matrix here, this matrix is called as stroma. Stroma. It's called as what? Stroma. Now, if you take one thylakoid, and we expand it, this is what we obtain. If you take one thylakoid and expand it, this is how it looks. This is how it looks. One thylakoid. That is a coin-like structure. Now, can you see in the middle of this thylakoid there is some gap here? Gap in the middle. That gap is called as lumen. The gap is called as lumen. The outer part is called what? Stroma. So here also stroma. This is the outer matrix, is the stroma. So whenever the protons are coming, so proton is coming from here, proton is coming from here. So can I tell inside the lumen proton level is increasing. In the outside there is low H plus, that is H plus is reducing. So that creates a type of proton gradient. Inside the lumen protons are increasing. Outside the lumen, that is stroma, protons are decreasing. That is creating a type of proton gradient. So what is the answer? It is between stroma as well as thylakoid lumen. Thylakoid lumen. Now, loss of biodiversity occurs due to growth of carrot grass called as Parthenium species. This is an example of. Again, students, ecology questions are going to be very easy for all of you. Ecology questions are going to be very easy, and questions in the ecology are going to be directly from NCERT. Directly from NCERT. So if you look at this paragraph right here, oh, it's not given here, okay. So this Parthenium is nothing but your carrot grass. You might have seen on the roadside, no? We call it Congress Gida. We call it Congress Gida. In Kannada, we call it Congress Gida. But that is a grass which is growing on the roadside. We usually think it is a weed. We don't take it and we just throw it out. right? That is called as Parthenium. Now that came into our country because of some transportation, illegal transportation. It came into India with the help of some rice, right? Some rice. So what is the answer? It's going to be alien species invasion. So it came as an alien plant. It came to India, and it is growing everywhere now. That is alien species invasion. Now, the egg apparatus is made up of 2 dash and 1 dash at dash end of the embryo sac. See here, if this is your embryo sac, this is the embryo sac, this is your egg apparatus. This is your egg apparatus. Egg apparatus is made up of 2 synergids, 1 egg cell, and it is found at the micropylar end. It is found at the micropylar end. Can you see? It has synergids, eggs, and it is found at the micropylar end of the embryo sac. So option number 4. Option number 4. Students, I know all these questions are little complicated, and if you really want to solve all these questions, you need to have certain level of concept clarity and apply those concept, and right. And if you want to know a harsh truth, all these questions are actually more difficult than NEET questions. Some of the questions are actually more difficult than NEET level questions here. So you need to know the concept, but I would suggest you to go for such easy questions such as from ecology. So study ecology completely, study the cell chapter study completely, and the photosynthesis in higher plants, that chapter. These 3 chapters focus more along with genetics. So these 4 chapters you need to focus more, and nothing can stop you from scoring marks. Just 4 chapters. Rest of the chapters, very few questions. If they come also, you can leave them. But if you have a lot of time and you have already covered many chapters, then go for every single chapter. But if you are starting biology for the first time, then go for these 4 chapters: genetics, cell, ecology, and as well as a little bit of reproduction. Okay. So with that being said, I will take off right now. Yes, and I will see you in the next class probably. And all the best to all of you. Do well, and bye bye.